Yeah, welcome to the opening keynote session for this fourth and last day of the Symbiote Expo 2022 conference. We're privileged to have Diana Thomas, who's on the faculty at the United States Military Academy at West Point, New York. Diana is a remarkable bundle of energy. I first met her in the last century, the late 90s, when she came as a brand new, a recent PhD to do a 3-year postdoc teaching position at West Point, and she became Army Green. She embraced the culture, ran with the cadets, and enjoyed her life there. Although, as we all find out when we go to an institution that has lots of rules, there are things that get in our way at times, okay? So then she went off to Montclair State, and we were fortunate enough at West Point to lure her back. And she's been a dynamo in the department. There are about 70 plus faculty, all in offices along the long corridor, and you can meet with them and talk with them in the hallway. And if Diana gets you and a colleague in a room for more than 10 or 15 minutes, you're probably going to wind up writing a paper with her, submitting a grant, or giving a talk with her. She has this ability to draw you in and engage you. And hopefully that's what this talk is. It'll share with you some activities that she's involved in based on anything from obesity to just fitness, and this has to do with diets. She's the recent recipient of a National Institute of Health grant, and for those of you who are in mathematics like I am, you ought to know NIH means lots of funding. You take your typical math grant and then you multiply it by 2, and then you add 2 zeros to the right, and you get a rough estimate of what the NIH is capable of supporting. And we're glad that they've decided to support Diana as a co-leader. So I'm going to turn the program over to Diana, and know that even though if you have questions during this, you can ask them in the chat. We're going to have another hour after this to continue conversations if you wish. So Diana, there you go.
Thank you. So I'm actually not a Zoomie. So if there are questions in the chat, if Brian—
I'll look at them, yeah.
And just interrupt me.
Okay.
So I thank you, Brian, for that introduction. And just like, I was wondering who that person was. So yes, to be fair though, when I first came to West Point, I was a bit more wild than I am today. So kids kind of have a way of calming you down. So there's been a convergence, I think, with rules and myself, just because I don't have a tomorrow anymore. Once you have children, you don't have a tomorrow. You only have today. You have to get it done. And so it's a different world. This is my disclosure, but I do want to add a couple of things to what Brian just mentioned. One is that this NIH grant is a West Point NIH grant. So yes, I did writing, but I had to make a case that we get this this grant. It's what we call a U54 Center grant. And with that, we have 6 different positions. So we're hiring with this funding. So if you know a recent grad student that is getting their PhD and would like to come work at the military academy, it's actually funded for 5 years. So it's a pretty long time. And so it's because of rules and regulations, I can't actually put in the ad that it's a 5-year grant because the DoD has some requirements and red tape. So that it's not in the advertisement, but it is an NIH funded position. There's several of them. So just ping me, send me an email if you have someone who's interested. And I'd love to have our community here apply for and work with us. So actually, my first little slide here is about Brian. So Brian has touched so many lives, and he gave that outstanding introduction for me. But as anybody who knows Brian knows that Brian really is what we call just top-notch mentor. I don't know anybody like Brianwinkle. Brian met me back in 1996, and he has written every single letter of recommendation for me my recommendations for my first job, my recommendations for my second job, my recommendations for tenure and promotion. I think he wrote my recommendation for full professor. He wrote a letter of support for my MAA New Jersey Distinguished Teaching Award. He wrote my letter of support to come back to West Point the second time. And he's not just doing that for me, he's doing that for a lot of us. When we go and compare notes, you find that Brian has stayed involved in our lives. There's 70 plus civilians that have been at West Point, and he knows all of us and he connects us to each other these days. So he keeps all this in his head, and he keeps it in his head because he loves doing it. Otherwise, I don't think he might just be a machine and keep it all in his head somehow otherwise, but I believe he really, really loves doing this. And so I am a product of Brianwinkle. So with that, I'm going to start with the unpleasant question for all of you. And I think it's really interesting that this is a food picture, but I'm going to ask you a food picture, a food question. I'm just going to ask you, since everybody just came from lunch, I thought this might be relevant. What did you eat over the last 24 hours? See if you can list everything. And I'm not going to use this data for anything. I'm actually just using this for the purpose of getting you to think about it, because it's easier just to ask, instead of asking a rhetorical question, to ask you a question that you can answer. And I'm looking for my chat box here. So how do I find that? Up at the top, here we go. So to everyone, I'm going to put a poll everywhere response and actually have you type it in. I'm not going to share it with everybody, just to get you to get a feel for it. You can put it in the chat if you want to, or you can put it in poll everywhere. So last 24 hours, try to think of everything. If you feel comfortable putting it in chat, you can put it in the chat. See if you can type out everything you had over the past 24 hours. See, I might have trouble with this. I cooked, so I ate little bits and pieces as I cooked, and I baked, and I had to taste what I baked. So I ate a brownie cookie thing. So I ate oatmeal for breakfast. That's measured. I don't know how long this takes everybody, but I think I would have trouble putting this together quickly. So with your hand raise, how many of you that are writing and trying to recall what you ate, say you're fairly certain that this is what you ate over a 24-hour period? Raise your hand if you're pretty certain that you have this correct, and try to put the amounts too. Like, I had 1 cup of this, or this much of this piece of bread. I had half a slice of bread. I had a 1/4 cup of skim milk. Definitely, it's a long list for me. I'm a grazer. Don't worry, Ruth. I am in the field. I have actually no problem having sugar. I've written a couple papers on, I can't find the evidence actually that sugar is going to cause anything, maybe tooth decay if you have excess sugar. So, all right, so we have hands raised that are people are fairly confident. Some people are fairly confident. Let me change that question. Let's suppose there were no statistical concerns whatsoever about covariates and bias samples. How confident would you be in your data that you're providing that could be used to make public health recommendations, or included in studies to make public health recommendations, or to design clinical trials so that we can actually use data like this to inform participants what they should be eating, or maybe inform people like, blueberries will make you live longer, so that we can derive scientific conclusions from the type of data you've provided. How many of you would feel like this data that you're providing is solid enough to make those kind of recommendations? Right, I see hands going down. Okay, so a few of you are kind of saying, okay, if I had known, maybe I would have been more precise. What's surprising is that even if you know that you're in these studies and you're going to be asked how much food you're eating, the accuracy goes down. And we have some evidence, some interesting evidence. There's really nice objective methods to estimate how much people are eating or what they're eating. There's a really neat sensor that sits on your jaw right behind your ear, and that sensor detects chewing and swallowing. So the engineer that built that device, it's tiny. It actually is barely visible, and you can see chewing and swallowing in participants, and they have self-report that is supposed to match the chewing and swallowing. And you see lots of chewing and swallowing without self-report. And it looked like to me that the self-report was put down after the fact, just like students that might be putting their homework in at the very last minute. People are generally more likely to keep records and turn them in when they're about to meet the clinical researcher. That should make sense to us as teachers, that this is how people behaviorally operate. But there was a fascinating study where all the researcher asked participants to do is write in a journal, write something, just write the date in the journal. And the journal itself was receptive to light. So when you open the page, light hits it and timestamps when you open that page. And what you found is people recorded that they were supposed to open it every day, but they made their records right before they met the researcher. I'm sure this doesn't sound surprising to us as teachers. So I think that's, you're talking about recalling what we're eating, that, even if we're given time, we probably will be filling that out at the last minute. And, when you look at data like the chewing sensor, what I see is myself. Busy people, they're busy people. And this is a volunteer, you're recording something and you're probably going to turn it in at the very last minute. However, scientific conclusions are being drawn from this data all the time. So when you read reports about sugar or different kinds of food or nutrients, they're based off of self-report. And what we just did was called 24-hour recall. There are different kinds of methods to collect this data, but you'll find that the methods correlate with each other, but do not correlate against objective methods. So what are those objective methods? One of them is mathematical models, and I'm going to get into that shortly. The lead author of this article is Dale Schuller, who has developed a chemical isotope that can be used through a urine analysis in free living humans to estimate how much they're eating on a daily basis. So it's not a surprise he's the lead author of this, but this article was written in response to a CDC study that said, hey, we noticed people are eating less, but they're gaining more weight in the United States. And so immediately from our exercise we just did here, something which should kind of go, well, wait a minute. If they're eating less, they're actually saying they're eating less. It's the CDC and Haynes data, the nutrition health survey that's done in the United States on a yearly basis, and that has self-report. So they're saying they're eating less. It doesn't necessarily mean they're eating less. And so that's why we wrote this piece here. How do we know how much people are eating? Well, the doubly labeled water method was developed by Dale Schuller from the University of Wisconsin-Madison back in 1988. It's so late 80s in humans, and it's a chemical isotope which estimates how much carbon dioxide you've expelled. From there, we can estimate how much oxygen you've consumed, and to ignite a fire, you need oxygen. So from oxygen consumption, we can estimate how much energy you're burning. Now, if you're in equilibrium, that means your energy intake is equal to your energy expenditure, then doubly labeled water is an estimate of your energy intake. And we can estimate that because your weight is the same every day. If your weight's stable, we say, you're in equilibrium for your weight, then you should probably be eating as much as you're expending. So your expenditure would give an estimate for your intake. So when we test this on humans, when we use it on humans and compare it to self-report, we see this discrepancy. So up from 10 to 30%, and they're all underreporting. So they're all in one direction. You see underreporting of self-report. And people have tried to look at self-report and say, well, maybe it's because of this. Maybe I can look at covariates and kind of clean up that data. It is impossible to clean up that data. People have different reasons why they might underreport. And it's not all because they don't want to record the ho-ho. There's other reasons why people might want to, or it might just be an accident. You know, I'm just signing up at the last minute, which would probably be more likely what I would do. This is one of my favorite papers. I taught a class in modeling and nutrition, and I would always assign this paper. It was written by a colleague of mine, Steve Heimsfield. In this paper, he took individuals who say, well, I do not eat anything, yet I'm not losing any weight. And he used the doubly labeled water method. This is 1992. So the invention of doubly labeled water and application to humans just started around this time. And so he applied it to these individuals. And you can also estimate how much energy you're expending in physical activity with another measurement, another clinical measurement. I'm not going to get too much into these clinical measurements. They're pretty fascinating. If you want to get into this field, there's lots of openings for people in the field. So if you're a mathematician that wants to be involved, you do have to learn about these measurements at some time. But right now, I'm going to kind of push it under a rug. But really, the measurement, you can figure out how much physical activity expenditures the individual has. And what he found was not surprising. Individuals were overestimating their physical activity and underestimating their intake by thousands of calories. So they reported eating about 500 calories a day in the mean. He enrolled individuals that say they weren't eating anything, and then they claimed they're eating about 500 calories a day, and they actually were eating in the 3,000, 4,000 range of calories per day. So really fascinating paper. It's freely available online. So if you want to find it, and if you can't find it, just email me. I'll get it for you. But there's been this study has been replicated. You'll find in PubMed, if you do a search, you'll find lots of papers like this on the discrepancy between self-report and objective methods. So let me pause there for a second and ask if there's questions.
Yeah. Can you hear me?
Yes.
Ruth Utnage asked, I'm curious, does smoking impact oxygen expenditure and thus impact the results?
One more. So no, oxygen consumption it's an objective method. So even smokers, the way we record expenditure is by oxygen consumption, and that's been tested in clinics. They actually have in metabolic clinics, they have something called a metabolic chamber with little vents at the top of the room. It's just a room. It's like a nice hotel room, and the individual will stay in that room, and you can actually give them their provided foods. And so you know exactly what they've consumed by calculating every single detail of their food consumption. And then you also have their expenditure. So this has been validated inside clinics. But it's a great question. I don't know if you've seen those things that people wear on their face when they're on a treadmill and it's a stress test, and that is also a way they're calculating oxygen consumption to see how much energy you're expending on the treadmill. So that's the same thing. But a great question. Other questions? Okay. All right. So just to give you an idea, these measurements, the doubly labeled water method is super expensive. It's about $2,000 a pop per person. So if you're enrolling 5,000 or 500 people, first of all, it's going to cost you a lot of money. So when you write your grant, you have to put that into your grant, and it's going to eat up your grant, and you won't have any funding to support your salary or your lab or anything else. And so people tend to not put doubly labeled water in their studies. And unfortunately, they revert back to self-report. Also, keeping people in the chamber costs a lot of money. Again, more money than doubly labeled water. So these measurements are super expensive. And so around the time that I came to the field, this was something that clinicians were really, really thinking about. Is there a cheaper way to do this? Can we use mathematical models instead to estimate energy intake? Because right now the status quo is self-report, and everybody seemed to understand self-report was highly unreliable, and it's not a measurement. It's like one of my friends says, you wouldn't build a spaceship and fly in measurements of that quality. So that's kind of where I came in. And the initial models I have do better than the one I have here. But one of the things that I'm going to tell you today is that simple is so much better. And sometimes it's a balance between simple and tractable. So you can build a model that's slightly nonlinear, that might need a Runge cutout to it. But if someone starts asking you for things like, I have 5,000 people enrolled and I want to estimate something from your models, and you're running these loops on 5,000 people, it starts, they start having problems with your code over nobody can use Mathematica or specialized code because they're not in math departments. They are clinicians, nutritionists, RDs. They need something like an R-Shiny app where they can access what you're doing. So partially you're writing these models so that you're addressing a question, you want to be accurate, but you're balancing that with, I need to actually hand this to someone who's going to use it. And so that second piece is important. So the model I'm going to show you, I call it a hamster model because it's running on one of those little wheels. And I think it does pretty well for how simple it is. So this model, the goal here I'm going to show, our goal is to first build a model, and it's going to be a differential equation model. And I want to keep it simple. So it's something that we can use in a calc class, actually. It's a great motivator. In fact, I think a great paper would be to write something about this kind of model used in the educational context for SciAm education. I think this would be a great paper for that. But you want to build a model to predict weights from energy intake, because that direction's easier. And explain why in a second. You can use the first law of thermodynamics to get there. Then you need to check, because this is the hamster model, and you need to check that that model actually does well. So you need data where you know what people were eating. So either they were locked up someplace and you know what they're eating because you provided the food, or you had doubly labeled water and these expensive measurements. Then you want to invert that model because you don't really want to know their weights. You want to feed the weights in and estimate the energy intake, that one holy grail that we're trying to get using other methods. So you need to invert that model. And that's actually annoying because it's not an inversion, algebraic inversion. It's an inversion of a differential equation. So what you'd like to do is take that inverted model, input the weights into the inverted model, and then predict energy intake from that inverted model. And then you have to check again that this inversion actually didn't mess things up. So you have to compare it to actual energy intake. So again, you need data where you know how much people were eating. So I'll pause there. That was a mouthful. Questions so far? Okay, I don't see anything in the chat.
No.
Okay. So the model that we're going to use is Vincent Antonetti's model. As far as I know, this was the first model that used the first law of thermodynamics to predict weight. And he was an engineer at IBM, and he told me he went to a cocktail party. And I thought, oh, we just call that a keg today. We don't call that a cocktail party. Well, they probably call it something else. Kids probably call it something else. But he went to a cocktail party. They were talking about Weight Watchers. They had used some rule of thumb. And he said, I think I can do better than that. And he approached the women that were talking about Weight Watchers, and he talked about, hey, I think I can use the first law of thermodynamics. And they looked at him and said, you're at a party. We don't want to hang around with you anymore. But he went back to the IBM where he worked, and he developed the first equation that I know of, differential equation model for predicting weight. And you see the big computers in the back. I thought it was neat. So how it works is you start with the first law of thermodynamics in humans, which boils down to what we call the energy balance equation. So what that says is it's the rate of energy stores is equal to the rate of energy intake minus the rate of energy expenditure. Don't let media confuse you. This is the first law of thermodynamics. It holds. So this is where we start from. So you'll hear, well, if you're on a low carb diet, it does this. No, it holds. And it's been, people have validated this. They call it validating, but it's really a physical law. And so they're just showing that the physical law works. Their clinical measurements hold up. So this has been tested in clinics where they have people in residence and they give the food so they know energy intake. They know energy expenditure because they're measuring it through the vents in the top of the metabolic chamber, and they know how much you weigh every day. So they know fat mass and fat-free mass and all the energy stores. And so you can actually experimentally validate this, and that has been done. And in the United States, we use calories. Kcal is the calories on the back of food packaging. 1 calorie in the back of food labels is actually a kilocalorie. So we use kilocalories per day. In Europe, they use megajoules per day. But you have an energy measurement, which is important for showing students a model like this, because you want them to hold to unit analysis. I'm dating myself. I dated myself when I said that I met Brianwinkle in 1996, but Matrix was like the movie when I finished at West Point. I show this to students. They don't know the movie anymore, but my kids were always fascinated by the baby batteries. The human body is an energy store, and that is what we're going to slice and dice to see what parts contribute to energy in the human body. So this is our human body broken up into pieces. The part that we worry about at the belly, that's called fat mass. And fat mass can be measured with what we call dual energy X-ray absorptiometry. I know that people think of skin folds. Skin folds are random guessing, or models with a waist circumference do better than skin folds. It's a trash measurement. You can also use bioimpedance. That's when you step on scales and it gives you an estimate of your fat mass, your fat mass. That's using bioimpedance. It's got quite a bit of error. People can do it fairly well, but still it's the best. Your MRIs and dual energy X-ray absorptiometry, DEXAs, are state of the art. And that's what the CDC uses in their national survey. The other parts are fat-free mass can be broken up into other pieces. Glucose, that's your glucose in your body. That's when they give you a glucose test. That's what they're measuring. Glycogen's what's packed away in the liver. Then you have proteins. That's your muscle and there's other parts of your body that are protein. And then there's everything else, water and bone. This part here, the yellow is fat mass and everything else is fat-free mass. So, and it's typically measured in kilograms. Different modelers choose different parts of this and cut-ups of this. So I'll show you in a second. Antonetti chose everything. Energy expenditure, we think a lot about physical activity, but the biggest part of the energy expenditure just goes to running our body. And that's called resting metabolic rate. It has some other names, but I always like to say when my husband's on the couch, he's actually working very hard. He's just running his body, and that's the hardest work. Your students, when they're sitting in class, they're working really hard, even though they don't look like they're working hard, just running their organs. You also have spontaneous physical activity. Most humans move and fidget. I've made everybody self-conscious here because they're moving around and they're going to stop moving because I just said something. But that's a small sliver of the pie. And then there's the thermic effect of feeding. That's the cost to eating your food. People worry a lot about this. They say, oh, don't eat at night. Your metabolism slows. Don't worry about it. It's just a small, tiny sliver of this gigantic pie. So those are your components of energy expenditure, your energy out. And again, it's a modeler's choice how they're going to divide up and slice and dice expenditures in a model. So back to here, what Antonetti did is he said, okay, I'm just going to take the baby battery, the whole thing, and let that be my energy store. Obviously, as mathematicians know, that's in kilograms per day. If I want to look at weight, the change in weight per day, Wrong units. I just told you it's calories per day. So the conversion constant came from early data in the 1950s and 60s that estimated how much a kilogram of weight is as an energy store, and it's about 7,700 calories per kilogram. And this is what you want students to do, right? They think you're boring when you do that, but it's really, really important to do that. And so now we have the right units. So that's energy stores. I'm just going to assume that fidgeting is close to 0 and throw it out. I didn't assume that, Antonetti did, but he has here in the pieces, 10% for of energy intake is an assumption that he made for how much is going to just digesting your food. That's been tested in the clinic. He made the assumption that physical activity is a direct portion of your weight. The heavier you are, it takes more energy to move your body. And resting metabolic rate, there's all kinds of fun equations out there for resting metabolic rate that's measured directly in labs, and they have tons of data that's actually freely available. So if you want your students to write regression models, this is great regression analysis. But I used Mifflin St. George. He used a nonlinear equation, and I told him, hey, we can use a linear equation here. And guess what? The whole equation becomes the hamster model. So we used something that's directly proportional to body weight, and the rest of it, you input these things in, it's just a constant. Non-mathematicians find that very disturbing. Like we take all these non-state variables and we collapse them into a Greek letter, and it has to be Greek. So it's the first point of confusion, I think, when we talk to a non-mathematician. But here we go. Rate of energy stores, I have this guy. Rate of energy expenditures, I have this guy. And I kept my EI in there because I really care about that controllable variable energy intake, and the state variable is weight. Just so you can see, this is what Mifflin St. George looks like. Covariates are height in centimeters, age in years, males and females. You have, it's a covariate actually, but it's usually on the web as two separate equations, one for males and one for females. So here's what it looks like to me as a mathematician. My state variable is up here. Everything else is a parameter. EI is really a parameter that I can input in here. And this is something technically a student at the end of a calc class should be able to solve with the integrating factor. So those rates of decay equations that we show them, I think they find this much more interesting. So I like to tell the students, a differential equation is an equation that involves the state variables and its derivatives. Nothing different than an algebra equation, well, somewhat different. I'm still trying to solve for something. It's just a whole trajectory I'm solving for when I solve this. What's neat about this equation is I can actually solve this. And so I'm not going to go into the solving of this. Everybody here knows about this, that we've taught this. But I need data now. Once I can solve this and I have data, I can actually see how well this hamster model does. So there, it's interesting to talk about, let me see if I have time here. Yeah, I have a little bit of time. It's interesting to talk about studies. So studies in the old days, in the long past, were just 10 people enrolled, 10 males enrolled in a study at a college. They were kept in-house, food was given. Then in the 1960s, 70s, and 80s, you started with more NIH-driven models for running clinical trials. And then it became, if you look at NIH's website, the institution owns that data. But it's gone further than that. It became that the clinical researchers own that data. So getting data became really, really hard. You do have publicly available data like Pew Research Center and things like that will put out their own data. But to get a targeted, like let's say I said, I want data that includes doubly labeled water, includes body composition, I have to actually go to those clinical sites and ask for that data. And most people don't want to share that data, because if you get that data, then you could write a paper on that data and you may or may not include them. And so that fear of losing that data is what keeps people from giving that data up. Around about 10 years ago, the NIH moved to a different model. They have these consortiums. Now NIH is moving to So this is a multi-site consortium. Their data is publicly available. So all of this data is in CSV files that you can download if you look up this study. And so now this was a push away from the investigators own the data, but they were allowed 2 years to publish anything they wanted before anybody else could access the data. And then the data also was not individual clinical centers. All these different centers gave their data to a center, a data center, and the data center is the one that analyzes the data. It was a biostat department at Duke that did that. And so, so here what you're seeing is that a place where you can actually download data for your students if you want to play with weight loss data. It's a 2-year weight loss study. But now they're moving to even something more. MotorPack was the first one, I think, that NIH did, where they did a multi-site consortium like this, but the data went into a repository. So I think the people, I'm not sure how they did the publications, but the first people that published on MotorPack were inside MotorPack. The one grant that I have, the Nutrition for Precision Health, it's going into a database called All of Us. And that All of Us already exists. It's a diverse database, and we're adding to that database, and anybody can access that data as long as they have an institutional account. So you just have to get a sign off. It's pretty easy to get. You sign on to these rules, and then you go into this database, and you can write our code inside the workbench. You never get to see the data. They'll tell you have 20,000 participants that satisfied this cohort, and then you work with the data inside the workbench, and you get the summary reports or whatever you want. So it's a very interesting way of getting data. But this is the data that that came from Calorie that I used, and their weight loss program. So energy expenditure by doubly labeled water is not enough. You need to also know changes in energy stores. So this change in energy stores comes from the literature. So I'm not going to get into that. This is how they test the first law of thermodynamics during weight loss. So let me just move on to the graphs. This is the actual versus predicted. Hamster model's not doing bad, considering that it's something that a calculus student at the end of the semester should be able to do, at the end of integral calculus, should be able to do. R squared is 0.87. You do see scatter, which is expected. Hopefully, if you don't see scatter like this, you start to think that I fabricated data. This is the residual plot. You see that it looks like it's a randomly distributed between actual and predicted. Slight underestimation of predicted from the actual, from the model, but again, not terrible. Very close to zero in the average error, and most of the error stays close to zero. What's nice about this, I'm publishing this with a group at West Point, but what's nice about this is that you can actually solve. I said Greek letters. We do have Greek letters. It was not my fault. It was somebody from physics that used all the Greek letters, and I kept it. I'm blaming it all on him. But we can actually invert that and solve for energy intake directly as a formula. So that's what's nice about it. You put in your beginning and end points into time, and you can actually get how much average energy intake was over that time interval. So now I can test how well it does to actual energy intake. And while the R squared is not something I would call home about to my mother, she doesn't want me to talk about math when I call her, but in general, I wouldn't call her about this. There is scatter, there's higher scatter the higher you get up here, but from where we came from self-report, I showed you those papers, that's what people are using. We're far beyond that. And it doesn't cost you anything because Jim Starling wrote a shiny app and you can just put your numbers in there. So it really doesn't cost you anything. This is the residual plot, just so you can see that there's no trend in the error and there's a slight overestimation of energy intake. So I think I'm almost out of time. So I'm going to go to the chat and see if you have questions. This is, if I really had to put all my collaborators, I would have slide after slide after slide. I work with these teams of individuals that are, some of them are math. I love being in a math department so I can still get help and talk to people about math problems, but I work with clinicians so I understand what their needs are. It's bumpy a lot of the times. I gloss over that, but you know, where I have a great idea, but it's actually not going to work out really well for them. And of course, Dale Schuller is one of my collaborators, and so is Vincent Antonetti. He's in his 90s. He's sharp as a tack. He's in Florida now, but he's professor emeritus from Manhattan, and a great collaborator. So I will stop there.
Diana, I have one question, and that is, how did you get into this kind of application of mathematics, and when did it occur in your career?
So I gained a lot of weight after West Point. I used to play a lot of basketball and not eat lunch because we'd go play basketball in Hayes Gym. And so once I stopped being at West Point and had a more sedentary lifestyle, I started gaining a lot of weight. And when I started losing the weight, I started thinking about the energetics behind weight loss. But then I met Stephen Heimsfield. He was a global director of obesity at Merck. I met him at a conference. And he came up to Montclair State and
wait a minute. What kind of conference were you at that you met a guy from Merck Pharmaceuticals?
So there was a reviewer for one of the papers that I had that was with an undergraduate student on fetal growth. And he was at the NIH. And he invited me to this conference at the NIH. And Steve Heimsfield was wandering around at the lunchroom. And I asked him, hey, you look like you want to have lunch with us. And my student was with me and he ate lunch with us and we started talking. Most people in clinical fields don't know the difference between statistician and mathematician and engineer. They think of us all as the same. We're related, but we're not the same. So he was asking me stat questions initially. And eventually I said, okay, I give up. I'm going to stop telling him I'm a mathematician. I'm going to learn some stats. I really learned stats though at West Point from Krista Watts. That's what got me into the field and undergraduate research projects and my own personal weight loss. I was nearly 200 pounds.
No.
Yes. I wasn't photographed much in that time, but yes.
Any other questions? We're going to go a couple of minutes over into the next session. We'll start the next sessions at 2:05 because we had a little bit of glitch, but are there any questions now? And remember, Diana will be in room 3 to continue the conversation and maybe to talk to her about the details of these positions and what sort of arrangements there are. So Jean Marie wants to know, where would you start looking for ideas for projects with undergraduates in this area?
So that's a great question. The problems that I work on with clinicians are clinician driven. So most of the time it's almost that you have to understand what they're asking. And then I start giving those projects out to undergrads, but there's more problems than there are people to work on them. We're overextended even at West Point looking at some of these problems and they can come from everywhere. One plug I'll make is in the summer, we hold a conference, a workshop. It's usually a week-long conference, but now we have it remote and it's got asynchronous materials over 4 weeks with synchronous times on Fridays. And for a 4-week period, there's a NIH grant writing workshop for it. And there's people that you can plug in with that are on the clinical side. It's called the Mathematical Sciences in Obesity Research. And it really plugs you into all kinds of clinicians that have these problems that could be accessible to undergrads. And I actually have an affinity for mathematicians who are applying. So, and I'm a PI of that grant. So if you apply and you ping me and let me know you're applying for that, I get your fee waived and I get you in there. I want as many mathematicians in there as possible. I just feel as a field, we have a lot to offer and we're all often overlooked.
Okay. So that's going to happen next summer.
That happens. Yeah, that's happened every summer for 5 years.
Oh, okay.
And it's happening for another 5 years because we just got renewed. So we have written, it's really great. One of your speakers here, Anuj Moyabi, well, he was one of our participants.
Okay.
So Swati DeBroy, there's been many mathematicians who've come through that and have written papers in the field and written with nutritionists and obesity researchers. So
okay. So your email is Diana Thomas.
I'll put it into the chat.
Okay.
And we're also trying to get on the map for the grant we have right now has 3 pilot projects, data science directed. And those 3 pilot projects are $200,000 each. And I have talked to Michael Pearson at the MAA about the possibility of talking about those grants, pilot projects at math venues, because you get a little bit of inbreeding with these things, and fresh ideas, different population, and the fact that math people usually don't apply for and get NIH funding. This will get your foot in the door, and we are the ones administrating it.
Okay. Well, thank you very much, Diana, for engaging us. You can contact her about the positions and about the workshops. And if you have specific questions, I know she is always excited to correspond and answer questions. And I warn you that if you get too close, you'll be writing a paper with her. And that's a good thing, but I'm just telling you, this is going to happen to you. So again, thank you, Diana. We're going to close this session and resume in a few minutes in the various breakout sessions in rooms 1, 3, and 2.
I got more questions here. So if somebody can, anybody like Victoria, if you email me, I'll send you back the link for the summer program.
Okay.
Thank you.
Thank you.